And thank you to everyone around the world for joining us for this week's lab meeting. My name is Kristen Aboud. I am the science editor at the Human Vaccines Project, and I will be your moderator today. After 2 deadly years of the COVID-19 pandemic, the Omicron variant continues to spread around the globe. Some public health experts are now suggesting that following Omicron, the combination of natural and vaccine-induced immunity may bring us to a new phase of the pandemic, while still warning that another viral variant is all but guaranteed. Today, we are joined by an expert in viral evolution who will help us understand how best to interpret the mutation of SARS-CoV-2. Before getting started with today's presentation, we would just like to congratulate the winners of the 2021 Michaelson prizes. These 3 awardees were selected from a global competition and will receive $150,000 research grants to pursue work that will advance human immunology, vaccine discovery, and immunotherapy research across major global diseases. These young investigators will receive their awards in a virtual ceremony on March 10th at 11 a.m., directly following that day's global COVID lab meeting. So we hope you will be able to join us for both events that day. Just one more note before I introduce today's speaker. The information presented today is currently under peer review, and we just ask that you not reproduce or disseminate the data in any way. With that, I am very happy to introduce today's speaker, Dr. Jessie Bloom, a professor at the Fred Hutchinson Cancer Research Center in Seattle and an investigator of the Howard Hughes Medical Institute. Dr. Bloom completed his Ph.D. at Caltech with Francis Arnold, studying protein evolution, and his postdoc with David Baltimore, also at Caltech, where he studied influenza virus. His lab uses a mix of computation and experiment to study virus evolution. Advances in genome sequencing have enabled rapid identification of new SARS-CoV-2 variant lineages, but a major challenge is understanding how mutations in these variants will affect antiviral immunity. In today's talk, Dr. Bloom will describe how new high-throughput assays provide data that can be used to rapidly interpret the consequences of SARS-CoV-2 mutations. During the presentation, please send me your questions using the Q&A function in Zoom. I will ask our speaker a broad selection of your questions after the presentation, and we will have about 25 minutes for discussion. With that, it is my pleasure to now introduce Dr. Jessie Bloom.
Thank you. Can you hear me okay?
Yeah, sounds good.
Okay, thank you for the kind introduction, Kristen. And by the way, thank you for the introduction, but people are free to reproduce or whatever they want from my talk. I'm posting the link to my slides in the chat. You're welcome to do whatever you want with it. The main thing I would caution you is I think we have all learned over the last 2 years, take any predictions about the evolution of SARS-CoV-2 with a grain of salt, including my own. All right, so I'm going to be talking about interpreting the evolution of SARS-CoV-2 in the context of Omicron. And these are the slides that I just posted the link to in the chat. And that link is right here. Okay, and before I start, I'd like to go through my disclosures. I'm on the scientific advisory boards of Flagship Lab 77 and Oncorus. I consult for Moderna. I'm an inventor on patents related to deep mutational scanning of viral proteins, and my lab has unfunded research collaborations with Vir Biotechnology. Okay, so the outline of my talk, I'm going to kind of break it into 3 parts today. First, I just want to talk about general principles of viral antigenic evolution and how those relate to what we've seen with the emergence of Omicron. Second, I want to talk about what might be the slightly niche, but I still think very important topic of epistasis and ACE2 binding by Omicron, and I'll explain what that means when I get there. And then finally, I want to focus for the majority of the talk on something that's probably of a lot of interest to people, which is antibody escape in Omicron. Okay, so first, let's just sort of take a step back from Omicron and think a little bit about viral evolution in general. So as I'm sure all of you are familiar, only sometimes does a virus's evolution lead to changes in its antigenic phenotype, or ability to be recognized by the human immune system. And two classic examples of this are measles virus and influenza virus. So these are both human respiratory viruses with RNA genomes that have very high mutation rates. Despite these superficial similarities, measles virus does not evolve to escape immunity. So in the old days, people got infected with measles at most once in their lives, and then a vaccine was developed in the 1960s, and it still works great today. In contrast, influenza virus does evolve to escape immunity. So people are infected about every 5 years, and the vaccine needs to be updated annually. So to sort of understand what we know about how coronaviruses fit in this spectrum, I first want to talk a little bit about another human coronavirus that's not SARS-CoV-2. This is the human so-called common cold coronavirus, coronavirus 229E. And this is a virus that's known to have been circulating in humans since at least the 1960s, and probably has been circulating in humans for much longer than the 1960s. So one of the first things my lab did when we were trying to understand coronavirus evolution is go and look at human coronavirus 229E, because unlike for SARS-CoV-2, we really have many decades of data we can go back and look at. So here what we've done is we've taken all of the known human coronavirus sequences isolated fairly directly from humans. So these go back to the oldest viruses up here in the upper left from the late 1970s and early 1980s, all the way down here to very recent viruses from the last few years. And we inferred a phylogenetic tree. So this is a timed phylogenetic tree, so time is on the x-axis here. And I hope one thing that you can appreciate that I'm going to come back to in a minute is how this phylogenetic tree has a ladder-like shape. So there's one major lineage, and then a lot of little short branches, which all end very rapidly, that come off this major lineage. And this just shows that every few years, there's a new variant of human coronavirus 229E that's sweeping through the human population such that the older variants go extinct. And so that's why all of the branches, except for this trunk branch, end. So to understand the evolutionary dynamics that are happening along this tree, we experimentally reconstructed the spike proteins from coronavirus 229E at 8-year intervals, starting here at 1984 and going all the way up to here at 2016. And then we used these spikes to generate spike pseudotype viruses in the lab so we could measure their ability to be recognized by antibodies. And we then did a really simple experiment. So we took an old human serum sample. So this is a human serum sample collected from someone in 1985. And we asked how well could it neutralize the coronavirus 229E spike from 1984. That's this point here. So a higher neutralization titer means you can neutralize the virus better. This is a reciprocal serum dilution that still neutralizes the virus. So this person can neutralize this virus that had existed a year before really well, probably because they'd been infected with something similar. But then if you look into the future from 1985, you can see that the ongoing evolution of coronavirus 229E erodes this antibody neutralization, such that this person's antibodies from 1985 can't recognize viruses, or can't neutralize viruses anyway, 20 years in the future. And in general, this is what we see when we look across lots of different human serum samples. In every case, when you look into the future from when the human serum sample was collected, those future viruses are not neutralized as well. So this is showing that this human coronavirus is evolving in a fashion similar to human influenza virus. It's escaping antibody immunity, and different from a virus like measles, which does not really evolve to escape that immunity. One other point I'd just like to make is that although the virus's evolution does erode everybody's antibody immunity eventually, it happens at very different rates. This person's neutralizing antibody immunity is eroded very rapidly by viral evolution. This is sort of a gradual decrease, and this is just a very slow decrease. And so I think this shows that even natural human antibody responses to coronaviruses can be very different in how susceptible they are to viral evolution. And I think this is an important thing to study because we'd like to have coronavirus vaccines that elicit antibody neutralization that's as evolution resistant as possible. And then the last thing I'd just like to point out is when you look to see where the mutations are occurring in coronavirus 229E, you see something that's very reminiscent of what we've already started to see with SARS-CoV-2. So the coronavirus 229E spike has the same basic structure as the SARS-CoV-2 spike. There's an S1 domain and an S2 domain. Within the S1 domain, there's an N-terminal domain and a receptor binding domain. And within the receptor binding domain, there are loops that contact the receptor, which in the case of coronavirus 229E is aminopeptidase N rather than ACE2. And when you look at the plot here, you can see that the highest concentration of mutations that occur over this many decade time span in coronavirus 229E is in the receptor binding domain, particularly the residues that contact the receptor. And then there's also quite a few mutations in the N-terminal domain. And this is exactly the type of distribution of mutations we see in SARS-CoV-2 variants like Omicron. The only exception is that SARS-CoV-2 variants also often have mutations that affect spike proteolytic processing, which is not something we seem to be seeing as much in a virus like coronavirus 229. So, okay, what does this mean sort of more broadly to now take a step back just from coronavirus 229E? Well, we know from nice theoretical work that's been done by a variety of people, including Trevor Bedford, that when viruses are under strong antigenic selection, they tend to have this ladder-like phylogenetic tree, just like I was showing for coronavirus 229E. And again, this type of tree means that at any given time, there's a little bit of variation, and there's one variant, the trunk lineage, which wins out and displaces the other variant. Then that happens again and again. So you get this ladder-like tree like I'm showing here. In contrast, viruses, human respiratory viruses that are not under strong antigenic selection, like measles, sort of have this more balanced tree because there isn't this immune pressure that's constantly causing one variant to win out over the others. And so human H3N2 influenza, for example, has a highly ladder-like tree, just like I was showing for coronavirus 229E. And in fact, this evolutionary property of human H3N2 influenza is the entire reason why we're able to sort of do these vaccine strain updates that we do for influenza. Although we don't know what it is, we're pretty much sure that in the future, there's going to be some dominant H3N2 variant. And obviously in times where there's different variants that co-circulate, as has happened in some recent years with 3C3A and 3C2A viruses, it makes the vaccine strain selection harder. And again, this is seen for most other rapidly evolving human respiratory viruses as well. So here's human coronavirus OC43. So this is another common cold human coronavirus. And here you can see OC43 has actually split into two coexisting lineages, which are now sort of evolving distinctly, but then each of those lineages is itself undergoing this ladder-like evolution, like we saw for coronavirus 229E or human H3N2 influenza. Something similar has happened with influenza B, is it's split into two lineages, which are then each themselves ladder-like. And again, this is why the influenza B component of the human vaccine can also be updated, although again, both lineages are needed. Okay, so this is sort of what, based on everything I've told you, we would expect to be happening with SARS-CoV-2. However, SARS-CoV-2 has really sort of thrown us for a loop because after the early fixation of this first adaptive mutation and spike called D614G, the SARS-CoV-2 tree has not so far been ladder-like. So the next variant has not come from the previous variant. So people may remember the first variant that everyone was talking about was Alpha. And for a while that was spreading very widely. And then we had this other variant called Delta, which is not descended from Alpha, which spread around the world. And if you'd asked me, and I think most other virologists or evolutionary biologists 3 months ago, we would have told you that we thought the next variant was going to come from Delta. Instead, the next variant has been Omicron, which did not come from Delta, but came from this other sort of long branch that we don't really understand how it evolved. So this is all very puzzling. And so far, SARS-CoV-2 evolution has lacked a lot of this sort of pattern of the next variant coming from the old variant, which appears to be a consistent feature of the antigenic evolution of other human respiratory viruses. Nonetheless, I would point out that the entire timescale we're looking at here is about 2 years. And when you go back to these trees for human H3N2 influenza or coronavirus 229E, they are ladder-like, but there are sort of non-ladder-like branches with lengths that span a few years. So anyway, we can't be sure what's going to happen with SARS-CoV-2, but I still sort of hypothesize we may eventually end up in a ladder-like scenario where the next variant comes from the previous one. So just to sort of the takeaways on the general principles of antigenic evolution of coronaviruses, other coronaviruses evolve antigenically, and they get mutations in the same regions as SARS-CoV-2 variants, especially the RBD and also the NTD. The only exception is SARS-CoV-2 variants also have mutations that affect proteolytic processing, and this is probably because the initial virus was poorly adapted to its furin cleavage site. And Michael Farzan has a nice perspective here talking about that. In addition, other antigenically evolving human respiratory viruses have ladder-like phylogenetic trees where the next variant often comes from the previous variant. And this property of the evolution is key to enabling vaccine updates as it provides some level of predictability. Namely, the next variant is usually descended from the last one. However, so far, SARS-CoV-2 has defied this ladder-like paradigm. Omicron isn't descended from Delta, Delta is not descended from Alpha, etc. However, I would point out it's still early. We're looking over short time frames. And right now we're seeing the combined effect of selection for antibody escape and for increased transmissibility. And I think transmissibility will eventually plateau. Therefore, despite having been wrong in thinking that the next variant would come from Delta, I'm still going to persist in saying that I still think that the evolution of SARS-CoV-2 is likely to eventually become ladder-like, although to take that with a grain of salt. Okay, so now I'd like to get a little bit more into talking about some specific properties of Omicron. So as I think everybody knows by now, Omicron is a remarkable variant because it has a lot of mutations, especially in its spike protein, especially in the S1 domain of the spike protein, and especially in the receptor binding domain, which is the part of the virus that contacts ACE2 and is also the part of the virus that's the target of most of the neutralizing antibody activity in human sera. And here's the structure of just the receptor binding domain, which is showing all of the 15 mutations in the BA.1 sublineage of Omicron. And so there's a lot of them, and a lot of them are up here sort of in the ACE2 contact surface, which is the top in this representation. There's also another cluster down here, which I think is quite interesting. So one question, and I remember one of the first tweets I read about Omicron was from Florian Kramer, who was just saying, holy cow, look at all of these mutations. Can this thing even still bind ACE2 after it's gotten so many mutations in the ACE2 binding interface? And in fact, the individual mutations in Omicron's receptor binding domain are predicted to have a net deleterious effect on ACE2 affinity. So for instance, in my lab, going back to 2020, we used this high throughput technique called deep mutational scanning to measure how all individual mutations in the original Wuhan HU1 receptor binding domain affect ACE2 affinity. And those measurements are here. And the key point is there are more negative numbers in this table than positive numbers. So in fact, 2/3 of the mutations in Omicron's receptor binding domain are measured to be individually deleterious for ACE2 affinity in the context of sort of the early SARS-CoV-2 strains. There are some like N501Y, which increase ACE2 affinity, but there are many more like K417N or G496S or Y505H that decrease ACE2 affinity. Nonetheless, despite Omicron having more mutations that individually decrease ACE2 binding affinity than increase ACE2 binding affinity, actually Omicron's RBD binds to ACE2 about 2-fold better than the early Wuhan HU1 RBD. Just as sort of a caveat, I'm referring here to sort of the RBD affinity in the context of the full spike. It's a little more complicated because the up-down conformation balance of the RBDs might be a little different, and that sort of indirectly affects overall spike avidity. But talking just about these RBD mutations, the Omicron RBD is binding to ACE2 better than Wuhan HU1. So how is that happening? The reason that it's happening is there's favorable epistasis among mutations in the Omicron receptor binding domain, such that some of these mutations bind to ACE2 better together than they do alone. So the measurements of the effects of the individual mutations doesn't accurately portray what happens when you combine them together. And in particular, two studies have shown that two of these mutations, Q498R and N501Y, can bind to really lead to a greatly increased affinity for ACE2. N501Y on its own leads to a fairly decent increase. Q498R on its own does not. And when you put them together, you get a really large increase. And that's probably largely responsible for the fact that Omicron is still able to bind to ACE2 so well. And it's also possible that there's epistasis among other combinations of mutations in Omicron, although this has not yet been fully experimentally elucidated. So I just point this out because Omicron represents the first major example of a SARS-CoV-2 variant of concern that has acquired sort of enough mutations that we are not able to sort of predict about what the effects of these mutations are going to be together from just combining the effects of the single mutants. And I think this sort of epistatic phenomena is going to become more pervasive as Omicron continues to acquire more mutations, or SARS-CoV-2, I guess I should say, continues to acquire more mutations. Okay, so now for the last part of the talk, I want to really focus on the property, or one of the properties anyway, that's been so notable about Omicron, which is its ability to escape from neutralizing antibodies. So I already talked about how Omicron has a lot of mutations in the receptor binding domain, which is the main target of neutralizing antibodies in human polyclonal sera, and how in fact many of these are kind of up on the top of the molecule where these antibodies often bind. So I now want to provide some really high resolution information on the effects of these mutations. So in our lab, we've been making a lot of use over the last 2 years of this yeast display system developed by Tyler Starr, who's a postdoc in our group, and Ali Greeny, who's a graduate student in our group. And the way that this system works is we just express the SARS-CoV-2 receptor binding domain on the surface of yeast, and then we can incubate the yeast with an antibody and see can the antibody still bind the receptor binding domain. And we can make very large libraries of these yeasts, such that, for instance, they contain all of the possible single amino acid mutants to the SARS-CoV-2 receptor binding domain, sort these yeast, and you do deep sequencing to really figure out how every possible mutation to this receptor binding domain affects binding by any given antibody. And so to give an example of what those data look like, here first are our data for a particular antibody. And so hopefully you're all seeing now something with a protein structure on it. If you don't see something with a protein structure, somebody unmute themselves and let me know. And let me just look in the chat and make sure nobody's, okay. Doesn't seem like anybody's saying they can't.
It looks good.
It's good. Okay. So here is the structure of this particular antibody called LY-CoV555, bound to the receptor binding domain shown here in gray. And so what we've done in these high throughput experiments is for antibodies like this, we've gone through every site in the receptor binding domain, and we've measured how mutations at that site affect binding by this antibody. And we've made the mutations for every measurement or every mutation, but just to make it easy to show, here I'm plotting just the average effective mutations at each site. So at most sites, mutations have no effect on the binding of this antibody. But there are a few sites like 452 or 484 or these other sites here where mutations do affect binding by this particular antibody. And in fact, this information turns out to be really important. So this particular antibody actually stopped being distributed by the, basically stopped being distributed almost as soon as it's gotten an emergency use authorization from the FDA. And the reason is that this particular antibody is escaped by both mutations at site 452, which are in the Delta variant, and by mutations at site 484, which are in Omicron and many other SARS-CoV-2 variants. So this particular antibody was basically useless almost as soon as it was improved. And so this suggests that one thing that might be a good thing to do if you were in the business of developing more monoclonal antibody therapeutics for SARS-CoV-2 is to figure out the possible escape mutations ahead of time, so upstream of spending very large sums of money taking the antibody through clinical trials, and then perhaps trying to choose an antibody that you think might be relatively unlikely to be escaped by the types of mutations that are prevalent in human SARS-CoV-2. As an example of an antibody that sort of has those properties, here's similar data for another monoclonal antibody. And so this monoclonal antibody is called S309. So again, here is the antibody in blue, and here's the receptor binding domain. And so this antibody was developed by Vir Biotechnology, and it's goes under the drug name of sotrovimab. And this particular antibody is not escaped by mutations at sites like 452 or 484. This antibody is really only strongly escaped by mutations at sites like 337 and 340. And just to give a sense of sort of the resolution of these data, when Omicron was first discovered, many people got worried that S309 would not work against it either. And the reason is that although Omicron does not have a mutation at 337 or 340, Omicron does have a mutation at site 339. And so if you look at it, 339 is right next to 337 and 340. And it looks like it's pretty much in the region where the antibody binds. So I think this is what people were looking at, and they were like, oh no, this antibody might not work. Nonetheless, these experimental data indicate that the mutation at site 339 should not affect the antibody binding much. And in fact, that's what's observed. S309 has less than a threefold impact for the mutations in Omicron. So again, I think this shows how this kind of data can be really useful for trying to understand the impacts of mutations on individual monoclonal antibodies. Again, there's the caveat is when you get a lot of mutations, there could be some epistatic effect. But so far, we've seen that more for ACE2 binding than for antibody binding. Okay, so those are two example monoclonal antibodies. And in fact, we've now generated comparable data on escape mutations for a whole bunch of monoclonal antibodies. So you can go to this website here, which is linked in the slides, and you can see this list of all of the different monoclonal antibodies that have been experimentally characterized in this way, so had all of their escape mutations mapped in our lab. So for instance, here's this antibody LY-CoV555, the Eli Lilly bamlanivimab, which doesn't work anymore. We can click here and we can see, yes, it has escape mutations at 452 and 484. And you can also see how that antibody sort of compares to other antibodies in this two-dimensional layout. And you can see that there's a lot of antibodies that sort of have similar properties to this one here. And then our lab has been doing a lot of this, but even more remarkably, the lab of Suni Shi at Peking University recently put out a huge study that applied this same approach to hundreds of antibodies. So if we combine these data sets, you can now go here and there are literally hundreds of different antibodies, and you can go through and you can kind of find any one of these antibodies, and you can click on it and you can see what are its escape mutations and sort of whether it's other antibodies that it's similar to in terms of its escape mutations. So this is really a wealth of information if you're interested in how any given mutation to the SARS-CoV-2 receptor binding domain is going to affect antibody binding. And I encourage you to play around. There's a lot of ways you can interact and explore these data. And also for those of you who care specifically about therapeutic antibodies in Omicron, there's a lot of data that's been published now. Seth Azos posted a really good Twitter thread, which has a pretty decent summary of it. And it's where I usually go if I'm curious about any given monoclonal antibody. Now, as hopefully you all know, most of us are not receiving monoclonal antibody therapeutics. Most of us are hoping that we're protected against SARS-CoV-2 infection because we have polyclonal antibodies that were elicited hopefully by vaccination or perhaps by prior infection. And just to make the point clear, a monoclonal antibody is a chemically defined species, such as might be produced by some company like Eli Lilly or Vir, and then it's administered as a drug. It binds to one part of the virus, and typically if the virus gets a mutation where the antibody binds, the antibody might not work anymore. But infection and vaccination induce our immune systems to make a polyclonal antibody response. And the polyclonal antibody response consists of an extremely large number of different monoclonal antibodies, each of which, at least in principle, can bind to a different part of the virus. And so if the polyclonal antibody response in practice ends up being balanced, so it has antibodies that are binding to many different regions of the key viral proteins, then it becomes very hard for the virus to escape this polyclonal antibody response. Because even if it gets a mutation that knocks off, say, the purple antibody, the gray and the blue and the green ones are still binding. Importantly, it's sometimes in practice, the polyclonal antibody response ends up pretty narrowly focused. So there's no guarantee that there will be this sort of breadth of binding. By the way, Kelly, I'm doing a presentation. And then so if polyclonal antibodies bind multiple epitopes, escape will now need multiple mutations. So for instance, we often group our monoclonal antibodies into these different antibody classes using a scheme developed by Christopher Barnes and Pamela Bjorkman. And the idea is that most neutralizing antibodies to the SARS-CoV-2 receptor binding domain bind to one of these 4 epitopes called class 1, class 2, or class 3. And then there's also a class 4 epitope where the antibodies tend to be less neutralizing. And so you would therefore think that in order for the virus to really have a major decrease in polyclonal antibody neutralization, it's going to have mutations in each of these epitopes. And so we can go and we can visualize antibody escape in the context of these different epitopes. So I'm now going to go here and show sort of all of the data. And then I'm just going to select individual antibody classes. So class 1 antibodies tend to target sites like 417, shown here, and 456. So in order for a virus to escape class 1 antibodies, it's going to have mutations at least some of these sites here. Class 2 antibodies tend to target sites like 484. Class 3 antibodies tend to target sites in the region from 440 to 450, like 444 and 446, and also site 346. And then class 4 antibodies target sites more in the 370s to 380s in the core of the RBD. And so this shows that we should really be thinking about mutations in terms of how many of these different epitope classes are hit. And so if we just combine all of the antibodies together, we can do something really simple where we just say where are the peaks of antibody binding? So where are the key escape mutations are going to be? And we can see there are positions like 484 and 417 and so on. So this gives us sort of a sense of the antigenic structure of the SARS-CoV-2 receptor binding domain, where mutations are going to have the biggest effect on polyclonal antibody binding. So to make that sort of information more actionable, we've developed this escape calculator, which just leverages all these high throughput experimental measurements. And so to give a toy example of how the escape calculator works, I'm going to show just a hypothetical mix of 3 antibodies, LY-CoV-016, LY-CoV-555, and REGN-10987. And for each of these antibodies, I've shown sort of where the antibody binds, conversely, where mutations escape it. So like LY-CoV-016 is escaped especially by mutations at site 417. LY-CoV-555 is escaped especially by mutations at 484, and so on. So if we imagine a mix of these 3 antibodies, such as might be in human sera, if humans just made these 3 antibodies, we would then expect the escape mutations to be sort of the average of these individual monoclonal antibodies. So this black line here. You can now imagine if the virus gets a mutation at site, let's say 484, that gets rid of this antibody, then if we subtract that antibody, the black line, so where the polyclonal antibodies are binding, changes because that antibody is not there anymore. And the same thing is true if we get a mutation that's say where this antibody binds, then it also falls out of the binding profile. So that's the basic idea of how we can just add up the effects of lots of different antibodies. And the key point is that escape mutations at certain sites are correlated. So for instance, both site 484 and 490 are antibody escape mutations. But if the virus gets a mutation in either one of them, it's going to escape the same antibodies and sort of have a similar effect. Okay, so now we can actually generate one of these escape calculators for all of these different monoclonal antibodies for which we have experimental data, like I was describing to you. And so this is showing over all monoclonal antibodies that have been experimentally characterized in this way, where are the key sites of escape mutations? And then we can further say, all right, let's say we make a mutation to site 484, which now drops 484 binding antibodies out of the profile. Now the new blue line shows where the remaining antibodies are binding. So you can see the next best escape mutation would now be at site 417. If the virus also gets a mutation at site 417, we can say what happens if those antibodies drop out. And you can now see the virus might want to get a mutation at 444, 445, or 446. And each time we make one of these mutations, we can say what fraction of the total antibody binding still remains. And so basically, we can use this monoclonal antibody data to calculate both the sites of likely future escape mutations and the total neutralizing antibody binding that remains after some arbitrary combination of RBD mutations. And just to sort of show, these predictions actually correlate quite well with neutralization assays. So prior to data being provided on Omicron, we made these correlations for sort of all other SARS-CoV-2 variants that have been characterized. And on the x-axis here is the fold change in neutralization for different variants, like this is the beta variant, this is a variant that just has S477N by various studies by the Iwasaki lab, the Sato lab, or the Ho lab. And then on the y-axis is the calculated binding score from the calculator I just showed. And I hope you can appreciate there's a good correlation here. And so based on this, we sort of predicted that Omicron was likely to have about a 25-fold decrease in polyclonal antibody neutralization from human sera. And indeed, that's kind of in the ballpark of what's been measured in different studies, although it depends a lot on which sera are used and which neutralization assay is used. So just to emphasize again, Omicron has this really extensive antigenic change. So Omicron has mutations at almost all of the key sites where antibodies bind to the original SARS-CoV-2 receptor binding domain. And really the most important antigenic mutations in Omicron are probably the ones at 484, the ones at 446, and the ones at 417. We can also ask, what are the mutations that escape the antibodies elicited by older SARS-CoV-2 vaccines that still neutralize Omicron? So this is taking just antibodies that still neutralize Omicron and now asking what are their escape mutations? And here you can see what they are. And from this, we can kind of identify what are the likely future sites of antigenic evolution in Omicron. And those future sites include 346, which indeed is already mutated in some fraction of BA.1 Omicron variants, site 444, and also sites like 486 and 487, which I think are less likely to change because they're highly functionally constrained. So anyway, I'd like to just end with some thanks. This work was led, most of the yeast display part I was describing, by Tyler Starr and Ali Greeny, who did a really great job developing that system. Rachel led the work with coronavirus 229E, and then Sarah, Kate, and Andrea all contributed to these studies in various ways that I don't really have time to highlight here. And then I'd also like to thank our many collaborators who've helped with this work. And overall, in the end, as you can see, I can't really tell us what's going to happen next with SARS-CoV-2 and Omicron, but hopefully I've shown some of the ways that we can try to think about this and at least be in a position to interpret and understand what might happen in the future. Thanks, and I'd be happy to answer any questions.
Great. Thank you so much, Dr. Bloom, for that very clear and fascinating presentation. We have a lot of questions in the Q&A, so let's jump right in if we can. We have a question on whether cooperation between different variants or lineages occur for SARS-CoV-2, as was shown by your lab for flu.
Yeah, that's a great question. So you're probably referring to Catherine Chu's paper, cooperation between two different influenza viruses helps their growth in cell culture. In fact, we think even for influenza, that phenomena is probably limited to cell culture. And for influenza, and probably also for SARS-CoV-2, I doubt there's a lot of cooperation during actual human infections, at least typical infections. And the reason for that is transmission bottlenecks for these viruses are very narrow. So even if a population is established within a host where there might be some cooperation between different viral variants, the transmission bottlenecks are so narrow that they would probably break apart that cooperating behavior. So although I think it's a really cool phenomena, and as you know, in that Eli paper from I think 2016, we described this really cool stuff that was going on in cell culture, I don't think at this point that it's likely to be sort of a major real world phenomena for influenza or also for SARS-CoV-2.
Okay. We have a question on whether you expect to see more mutations in T-cell epitopic regions.
Yeah, I'm not really an expert on T-cell escape, but I can make a couple of sort of high-level points about that. First of all, T cells are probably putting somewhat less strong selection pressure on the virus. And that's for two reasons. T cells mostly mitigate disease severity rather than block infection. And from an evolutionary perspective, the virus is mostly under selection to transmit, not really, what happens to your disease severity 4 days later. Second, although there is some antigenic selection in T cell epitopes and things like flu, it's much less than the antibody selection. And I think the other reason is because T cell epitopes are so different from person to person. So you do see a lot of T cell escape in viruses like HIV, which are chronic viruses that evolve in the same person who has the same HLA type and recognizes the same epitopes for a very long period of time. However, for respiratory viruses like flu or SARS-CoV-2, every couple of days or at least every week, they're transmitting to a new person who usually will have a new HLA type and will recognize different T cell epitopes. And that makes the evolution very difficult for the virus because the mutations that it started to accumulate in one infection that might help it no longer help it in the next infection. And so I think the diversity of human HLA types probably really slows down T cell escape from viruses that transmit very rapidly from person to person.
Right. That makes a lot of sense. We have a question on whether you think the increase in vaccination led to the development of Omicron's multiple mutations. And was it due to the increase of a polyclonal antibody response being more present in the population? And then following on that, what do you believe future outbreak variants will have in terms of their number of mutations? So we're back to asking you to make more predictions.
Okay, so first of all, whether vaccination contributed to the emergence of Omicron, I have no way of saying for sure, but in general, I'm pretty circumspect about our ability to control viral evolution with Omicron. With vaccination. I've heard it both ways. You hear people say that, because there were vaccines that put this pressure and this is why the virus evolved antigenically. You've also heard people saying because vaccines weren't distributed widely enough around the world, that led people for the virus to infect and that contributed to viral antigenic evolution. I think in practice, there's a lot of people out there, sort of regardless of whether vaccines are administered or not, a lot of people are going to have immunity because if they don't get vaccinated, they're going to get infected. And in practice, when we look at viruses like influenza, they've been evolving antigenically now that we have vaccines. But if you go back, 50 or 100 years before there were any vaccines, they were still evolving antigenically. So I think the reality for viruses like flu and SARS-CoV-2 is they're going to undergo antigenic evolution because the host population is going to acquire immunity from either infection or vaccination. And we probably can't really control the evolution by what we do. And so I think we should think of the use of vaccines for just the goal of ensuring that people don't get sick as much as possible.
Right.
As far as whether the next variant will be like Omicron, the emergence of Omicron is very surprising. I think the most likely explanation is it has something to do with persistent evolution in a chronically infected individual, but it's not known. And it's very surprising to see this virus emerge on this long branch.
Right.
I don't think that's going to be the norm. I still feel like the norm is going to be in the end, SARS-CoV-2 will kind of be like other respiratory viruses. And it will usually evolve in a more gradual way with variants being descended from other variants. But, like I said, I didn't predict Omicron was going to happen. So take that with a grain of salt.
Yeah, I'm sure that makes a lot of sense. So we have a question about what your work might tell us about whether BA2 compared with BA1, and is there any chance of BA2 escaping antibodies to BA1? Of Omicron.
Yeah, so I don't have a plot here that really clearly shows BA1 versus BA2, but so people who don't have the background, what people have generally been referring to as Omicron is the BA.1 sublineage of Omicron. There's another sublineage of Omicron called BA.2, and in some countries, BA.2 is now increasing in frequency relative to BA.1. They share many, but not all of the same mutations. Most prominently, BA.1 has a significant antibody escape mutation right here at site 446. This is a major site of antibody binding. BA.2 does not have that mutation. And then there are a few other mutations in the receptor binding domain. I'd expect that the neutralization properties of BA.1 and BA.2 will be a little bit different, but I still think they're going to be fairly similar. I'd expect BA.2 to have a little bit less escape from immunity elicited by early vaccines. I'd also think that antibodies that are elicited by BA.1 will probably still react reasonably well against BA.2, certainly much better than, let's say, Delta antibodies reacted against either Omicron lineage. And that's because BA.1 and BA.2 are not identical, but they share many of the same mutations. So I would be surprised if BA.2 caused major waves in places that had just experienced a BA.1 wave, because I would think the BA.1 immunity would have some protective effect. Nonetheless, BA.2 is clearly spreading in some places relative to BA.1. And another possibility is that BA.2 could be inherently more transmissible for reasons unrelated to antibody escape. So those are my hypotheses, but none of these things are really confirmed by experiments at this point.
Right. Okay, thank you so much. We have two questions on the mutations in non-structural proteins and whether you think there'll be any resistance to some of the recent antivirals for SARS-CoV-2.
That's a great question. And I'm just going to have to say, I don't feel like I'm really informed to answer it. It's an important question, but I'm not the right person to answer the question.
Absolutely. No, completely understand. So also we have a question here on the escape calculator. And would you be able to use that to determine which monoclonal antibodies would be best for on a patient level?
I don't even think you'd need the escape calculator for that. I mean, you could just, so in principle, you could imagine a scenario where someone gets infected with a virus and they sequence it, and then they say, all right, this antibody will work against it. And this was particularly valuable during, let's say, the time frame when Omicron and Delta were co-circulating, which is still happening maybe a little bit, but Omicron's basically displaced Delta. Because there's a lot of antibodies like the Regeneron cocktail that work against Delta, but don't work against Omicron. I'm not a clinician, but my understanding, however, is that the practical timeline of how long it takes to sequence these viruses, as well as the availability of drugs, probably makes that just not feasible, because these antibody drugs need to be given pretty early on to be effective, and the time lag for sort of sequencing the virus and everything would make that too long to do on a per patient thing. Nonetheless, you could imagine a scenario where there are different variants co-circulating and the antibodies work against some, but not the other. And just by looking at community level thing, you could try to guess what a given patient has. So I think there was probably a window of time when Delta and Omicron were both prevalent where this would have been a useful approach. Right now, Omicron is so prevalent everywhere that probably we basically just need to use antibodies to work against Omicron.
Right. So we have a question on whether you think vaccine manufacturers should move to produce Omicron-specific vaccines if there is a suspicion that the next variant would be Omicron-derived.
So with the caveat that I'm obviously not in a position to make statements about the cost of different approaches, in terms of money and all of the need for clinical trials and things like that. I think at this point, and also that I would again acknowledge that I was wrong in thinking the next variant would come from Delta. So given all those caveats, I nonetheless think that based on what we know now, there's a good chance the next variant will come from Omicron, and Omicron definitely has enough antigenic mutations that if that's the case, I think we should start using an Omicron-specific vaccine. For Delta, that was not a very strong case because the original vaccine still worked quite well against Delta. So in my view, it would be very prudent to be pushing Omicron-specific vaccines forward. That said, how far forward they need to be pushed before certain of these things is hard for me to say, because I'm not in a position to make the cost-benefit analyses there. But I think from a scientific perspective, there's a lot of reasons to think right now it'd be a lot more beneficial to be getting an Omicron vaccine than the current vaccines. Although it's also still established that boosting with the current vaccines does increase your Omicron antibody levels to a reasonable degree.
Right. Okay. Thank you very much. We also have two questions on if you could comment on what is unique about measles and why doesn't there seem to be the same immune pressure on that virus?
Yeah, that's a great question, because you would think measles would get a huge advantage from that. So from escape. So there's sort of two hypotheses out there. One is that the properties of the measles surface proteins, the measles hemagglutinin protein and fusion protein, is such that just it can't really get mutations in the regions that antibodies bind without messing up the function of these proteins so badly the virus can't transmit. So it's just evolutionarily constrained. That's one hypothesis. The second hypothesis, and there's some recent evidence for this, is that the antibody response to measles appears that the neutralizing activity is very balanced across many different epitopes in the measles surface protein. And that sort of goes back to this sort of image I was showing here, where if the neutralizing activity really targets many, many distinct epitopes with about equal potency, that makes escape hard because the virus has to get a lot of mutations kind of all at once to really appreciably escape polyclonal antibody immunity. So I think those are the two most likely hypotheses. Measles might be more functionally constrained, and the natural neutralizing antibody response to measles may be more balanced across many epitopes than what we see for flu and coronaviruses.
Okay. So we have a couple of questions that are related to the evolution to Omicron, which was more transmissible but less severe. And if this is the next variant comes from Omicron, are we following that same pattern? I think you also said that we kind of maybe tapped out on the increase in transmissibility. So we have a few questions on that.
So first of all, I would not say, those are both good questions. I would not say that I'm sure that we've tapped out on increased transmissibility. The virus could get even more transmissible. But I do think, just from a fundamental evolutionary perspective, eventually transmissibility is going to plateau. So a virus has essentially an infinite space for antibody escape, because it can always get a new mutation somewhere where an antibody binds, and the antibodies bind there, and that gets a mutation there, and this process can kind of go on forever. Transmissibility, eventually it's going to plateau. When we look at other viruses that have stably been in humans for a long time, like measles or H3N2 influenza or things like that, there's not evidence they're continuing to get more transmissible every year. So I think eventually that will plateau. Whether Omicron is at that plateau yet or not, I can't really say. There definitely is good evidence that Omicron appears to be less inherently virulent than something like Delta, and there's speculation that this could be due to spike proteolytic processing, restricting the cellular tropism of Omicron a little bit. Hopefully, that will be a property that's preserved in future SARS-CoV-2 variants, although I can't be sure. As I think many people know, the virus is under very strong evolutionary selection for transmissibility, but the severity of the disease caused by the virus doesn't, in the end, matter that much to the virus's evolution. So it's certainly possible for a virus to evolve to become less virulent, but it's also possible for its virulence to stay the same or even get higher. And because that's a property that's not inherently under a lot of selection and probably just almost changes incidentally, it's hard to predict what's going to happen there. But certainly, I think in the long run, regardless of the inherent virulence, as more and more people have some level of immunity to SARS-CoV-2, and at this point, with both vaccination and infection, most people in the world have some immunity to SARS-CoV-2. I think that's going to cut down disease severity regardless of what variant people get infected with.
Right. Okay. That makes a lot of sense. Thank you very much. So we have a question if anyone is developing antibody escape maps for other viruses, for example, for RSV.
That's a good question. We're in our lab doing similar work for flu, and we've actually been interested in trying to do it for RSV. So I can't see you ask the question. If you're specifically interested in RSV, let us know. There's a lot of technical challenges, like the particular approach we used for SARS-CoV-2, yeast displaying the receptor binding domain probably would not work for RSV. Some proteins can be yeast displayed, some can't. But yeah, I think it's a great question. I think it would certainly be useful. Many things about science of respiratory viruses have advanced a lot with SARS-CoV-2, and I think applying some of those same approaches to other viruses would be really great.
Okay, thank you. So then we have a question. Could you have a mixture of antibodies that would induce a three-dimensional change that would basically incapacitate the ability of COVID to bind to ACE2 receptors?
Yes. So it's actually unclear how a lot of the antibodies neutralize the virus. Some of them just bind to the RBD and get in the way, so the RBD can't bind ACE2 anymore. So there's an antibody stuck in the part of the RBD that binds to ACE2. Although I'm not entirely up to speed and can't quite remember the details, there also are antibodies that bind to spike and cause some conformational conversion, which essentially kind of destroys the spike and in the end makes it unable to bind to ACE2, or at least unable to mediate fusion after it binds to ACE2. So there are some antibodies that work that way. So, the mechanism of how all neutralizing antibodies work is not well understood, but destruction of the viral protein by causing some conformational change has been shown to be a mechanism of at least some antibodies.
Okay. We have a question on how representative the RBD is on the yeast display system compared to the one on the native virus spike.
Yeah, that's a great question. So people usually have two reasons they worry about the yeast display system. One is that although yeast glycosylate proteins, the glycans are a little bit different. And so they always worry about whether the glycans in yeast displayed RBD are the same as in human or cell expressed RBD, and they're certainly not. However, we haven't really seen cases where we think that influences the results that much. The bigger factor that I think affects the accuracy of our RBD based yeast display is we're just looking at the isolated RBD, but in reality, the RBD is part of this larger spike structure, right? Here's the whole spike, and the RBD is just one subdomain of that. And so the interactions between the RBD and the rest of the spike can have important effects. So for instance, I actually just was tweeting about this morning before the seminar. There's mutations at site 371 in the RBD that actually reduce neutralization by a fair number of different antibodies that don't actually bind to that site. And there was just a nice paper that came out that suggested that could be because those mutations put the RBD in a different quaternary conformation, the down conformation more in the spike. And so even though that mutation isn't directly escaping the antibody, it's kind of putting the RBD more in a position where those antibodies can't see their epitope as well. And that's not the kind of thing that we would see in our yeast display. We just totally miss that.
Right. So I think related to that, we have a question asking how you assess if a change in the RBD is advantageous or deleterious.
Yeah, we kind of actually try to avoid, that's a good question. We try to avoid saying beneficial or deleterious in terms of viral fitness, because the fitness of a virus, its ability to transmit in the human population, is an extremely complex property that's hard to measure in the lab. We try to think of it as we are measuring how mutations affect key biochemical properties of the RBD, such as its affinity to ACE2, or whether it can be recognized by our antiviral antibody. And those biochemical properties contribute to fitness, like we think the virus has to be able to bind ACE2 to transmit. However, the exact quantitative relationship between the biochemical properties we're studying and fitness and transmissibility is hard to know. So that has to be always kept in mind.
Right, right. And we had a question on whether your lab has had a chance to really characterize the BA.2 mutations. And if so, does it match with the sites of likely future change that you highlighted in your talk?
We have not. In fact, I have to say our lab has actually just kind of stopped trying to rapidly characterize new variants as they emerge, simply because there's so many labs doing that now. It seems like there's very little value added for us doing it as well. But we are looking a lot at the mutations in terms of what we know comprehensively about the effects of mutations from these deep mutational scanning experiments, and also what other people have done. As far as I'm aware, there's not yet any direct characterization of BA.2. As I mentioned, based on what we've seen in this high throughput yeast display, we think that BA.2 might have a little bit less evasion from current vaccines than BA.1 due to lacking the G446S mutation. But I think overall their antigenic properties are going to be similar, although not identical. Probably kind of like Delta was kind of similar to the original Wuhan H1, but not identical. I think there'll probably be about the same amount of difference between BA.1 and BA.2.
Right, right. So maybe we'll close with one last sort of question, which is, as structural biology improves, do you think that scientists like yourself will be able to visualize the actual immune system environment, or will it always be too complex to do that in a useful or predictive way?
So the immune system is super complex. We're never going to understand everything about it. That said, I feel like we understand a lot more about it now than we did, certainly 10 years ago, and honestly, even 2 years ago, when we started working on SARS-CoV-2. So I think that understanding is going to continue to improve. But I think as a scientist, it's always important to try to get better data and integrate it, but also remember that the real world out there, like transmission of viruses among people with whole immune systems, is really complicated. And we also always have to be circumspect about probably we're not measuring everything in experiments, and we probably never will.
Well, thank you so much, Dr. Bloom, for an excellent presentation, for really thoughtful answers to the dozens of questions we got. We really appreciate your taking the time to share this data with our audience and for answering all of the questions. Thank you so much.
All right, thank you.
I would also just like to thank all the attendees for participating in today's webinar and for submitting such great questions. We are fortunate to have such an engaged audience for these lab meetings. And I invite you to join us 2 weeks from today on February 10th Our speaker that day will be Dr. Felix Braden, Professor Emeritus of the Simon Fraser University, and he will be joined by Kira Neller and Brian Corey of the iReceptor Project. Together, they will be speaking about adaptive immune receptor repertoire sequencing data and how it facilitated a rapid response to COVID-19. And if you're interested in more research on COVID, please sign up for the HVP COVID Report, a biweekly newsletter that provides insights from experts around the globe and highlights the latest scientific articles and data. And finally, please visit our website and follow us on LinkedIn, where the full webinar series is available, including our exciting lineup of speakers for this year. And with that, I'll say thank you again for participating today. Stay safe, and we hope that you'll join us again for the next Global COVID Lab meeting. Thank you so much.